Hey guys, this is Mrs. Kraus, and in this video we're going to be talking about how mitosis affects the number of chromosomes in daughter cells. So, mitosis is the process that we use to make new cells that have the same number of chromosomes as the parent cell. We want to do this when we're creating new body cells, let's say to grow or to replace cells that were lost as a result of some injury. And an example of a body cell might be a bone cell, a skin cell, a blood cell, a muscle cell. So I've given you a little diagram here. Humans have 46 chromosomes in each of our body cells, and we're considering those cells to be diploid, which means that they contain two sets of chromosomes. In humans, each set is 23, so two sets would be 46. You get one of those sets of 23 chromosomes from your mother and one of them from your father. So we've got our diploid parent cell here with 46 chromosomes. When it goes through mitosis, it actually creates two daughter cells that each have 46 chromosomes as well. So they would also be considered diploid. So we're going to take a look at how mitosis is able to produce daughter cells that have the same number of chromosomes as the parent cell. Okay. So in our part 1 notes, we went through all the stages of mitosis, so all the stages that a cell goes through as it divides all the DNA within its nucleus. So this is a picture, a detailed picture of metaphase that the parent cell is going through. And let's say instead of having 46 chromosomes, the parent cell only has 4 chromosomes. That way we can draw it more easily. So I've shown my 4 chromosomes lining up at the center of the dividing cell. That center line is called the metaphase plate. And I've labeled my chromosomes. This is chromosome A, this is chromosome B, this is chromosome C, and this is chromosome D. If we look more closely, we can see that each one has a number 1 side and a number 2 side. So if we look over here, we see that chromosome A has A1 on the left and A2 on the right. A1 and A2 are supposed to represent chromatids, so identical copies of DNA that are joined together at a region in the middle called the centromere, and that makes up a single chromosome. So we're starting out with 4 chromosomes, and we would say that they're replicated, which means they have 2 copies of DNA, the 2 chromatids. So they're lining up at the center, and they are connected to the spindle fibers. The next thing that has to happen is anaphase in our parent cell. So when A1 and A2 get ripped apart, they kind of invert, so they look a little bit more like boomerangs, and they're going to be pulled to opposite ends of the dividing cell, along with B1 and B2, C1 and C2, and D1 and D2. So they're going to start moving towards the ends of the cell, and at this point, once they've been ripped apart, A1 and A2 are each considered full daughter chromosomes. They're no longer considered chromatids once they've been ripped apart. Okay. Now let's say my cells split down the middle like this and finish cell division, and my 2 daughter cells now entered their own interphase. So they're starting their own cell cycles. Now, something that I want to keep in mind is that our chromosomes would not be coiled up as chromosomes at this point. Our DNA would actually be uncoiled as chromatin, but I've shown them coiled up because they're a lot easier to see that way. So these are our 2 daughter cells that resulted from this mitosis. And it looks like in the nucleus we have A, B, C, and D in each of our daughter cells. So they still have all 4 of the chromosomes, A, B, C, and D. It's just they are unreplicated, so they do not have the other side of the X. So this is G1 of interphase, where the cell's just growing. But in the S stage of interphase, something important happens. During the S stage, which is called the synthesis stage, we're synthesizing a copy of our chromosomes, so we're replicating them so they now have 2 chromatids. So we're going to see that our daughter cells now have the exact same DNA that the parent cell did. We've got A, B, C, and D, but we have A1, A2, B1, B2, so each of our chromosomes is now replicated. Okay, so you can see that these cells are identical to the parent cell that was going through metaphase initially. It's got the 4 chromosomes and each with 2 chromatids. So we're ending up with these 2 daughter cells that are diploid. They have 2 sets of chromosomes just like the parent. And in humans, that'd be 46 chromosomes, not 4. All right, I hope this was helpful. And just as a recap, we were exploring how mitosis allows us to create daughter cells that have the same number of chromosomes as the parent cell.